Kia ora koutou, everyone. My name is Jay. My pronouns are he, him. So as Travis has already introduced me, I'm a scientist working for plant and food research. So today I wanted to share with you some of the lessons that we learned as a result of some funding that we got for a postdoctoral position at plant and food research, working on, title gives it away, PSA. And some of the lessons that we learned, some of the learnings that and technology transfers that we achieved as a result of that funding. I apologize in advance that this is a very science-heavy presentation. I welcome any questions after. So I'll jump straight into it. So PSA. Apparently, oh yeah. So PSA, Pseudomonas syringae pathovar. Oh, okay, all right. Pathovar actiniae is a pathovar of Pseudomonas syringae, a bacterial pathogen. You may all be well aware of this by now. Caused a devastating disease, bacterial canker disease in the kiwifruit industry, completely destroyed the current, then current gold cultivar Hort 16A, and some of the symptoms are on screen there. And so the PSA itself, pathovar, is divided into multiple clades. The one that came into New Zealand in 2010 is PSA 3, and it's the most virulent of the lot. But there are other clades that are out there, PSA 2 and PSA 1. It's also related to a mild pathogen of kiwifruit that's ubiquitous around the world, PFM. I won't be really talking about that at this point. Pictured on the right there, bottom right, are Hort 16A plants that we use in tissue culture. So these are plants that when we infect with PSA, after 50 days, these are the typical symptoms that you see. The symptoms kind of develop progressively up to 30 days, and then there's a rapid sort of switch to the plant dying and completely losing control. So we wanted to understand what the mechanism was behind this disease, and so this is a very critical fundamental research. There's a lot of research that has already gone into Pseudomonas syringae, less so at that point when I entered into PSA and its mechanisms. So, oops, sorry. So Pseudomonas syringae, on her journey of self-discovery, history of a pathogen, becoming all that she can be, we sort of look at what are the things that are essential for P. syringae to be able to be a pathogen. And so along this journey, I've indicated it with a red star, is the acquisition of a very complicated name, a type 3 secretion system. It's basically just a molecular syringe that pictorially are represented on the right there. So the blue lines are the membrane of the bacterial cell, and so what the type 3 secretion system is a set of genes that Pseudomonas syringae has acquired that allow it to build this protein system that can then inject proteins, other proteins, into the plant cell. So these proteins are effectors, as Travis has mentioned earlier. They are injected into the plant cell, and that performs a variety of functions. Through studying other similar Pseudomonas syringae pathogens, in this case tomato, the community, science community has identified several effectors that are key to causing disease. Most of these effectors are involved in suppressing the innate plant immunity. In plants, every cell is able to defend itself. It doesn't have a circulatory system like blood flowing through it the way that we do, and so it doesn't have a moving immune system flowing through it. So every cell defends itself. So it's, in a way, it's very good because then every cell can then stand up for itself. But that means that a bacterium has to come in, and every bacterium has to defeat the system. And so it uses the type 3 secretion system, injects these effectors, and the vast majority of them are involved in immune suppression. A small handful, so in the blue at the bottom there, HOPM1 and AVRE, the names aren't that important at this point, but they are effectors that are involved in making a wet epiplast. So they release water from the cells into the empty space within the leaves, and this pulling out of the water allows nutrients to flow out, and this is absolutely essential for the bacterium to be able to grow, divide, and replicate. And so it became very clear early on that this is going to be very important for a Pseudomonas syringae pathogen. And so this is when we sort of entered with PSA. This was all knowledge that we had. So with PSA, it has a large number of effectors, quite unfortunately, distributed amongst the different biovars or different strains of PSA. So the one we're primarily interested in is PSA3. So the way, so when I came in, we were interested in knocking out, so genetically removing each of these, either in groups or individuals, of these effectors to understand what contribution they played in the disease of kiwifruit. And so I'm not going to go into the details how we did the knockouts. I'm just going to hand wave and say we did a little bit of science magic, and we got a collection of these mutants. It took 3 years, but it was very rewarding, very easy for us. So we got a collection, we call it a library of knockout strains for individual effectors knocked out. And then we use a classic in vitro infection assay. It's basically those little plant tubs that I showed you earlier, a photo of. We, in a very complicated setup, we take the plants, we flood them with 1 million cells per mil of PSA, and then pour it out, and then wait, and sample the leaf tissue, grind it up, and look at how much bacteria is in there. And we also keep some plants left over for some photos, because everyone loves photos. So for the quantification, what we're looking at, I'll sort of depict it here. Travis put a similar version of this up earlier. So in every graph that I'll show you, the red bar represents the original PSA strain. So it has very high virulence, and that's depicted by the height of the bar. The higher the bar, the more PSA is colonizing the plant. And so the scale on the left is a log scale. So 2 is 10 times more than 1, 3 is 10 times more than 2. So it goes up very rapidly. This is exponential growth. So if you see a slight decrease between 9 and 8, that's 10-fold more. Two slots down, that's 100-fold less. So what we're looking for is when we knock out a gene, in this case, the HARC-C gene. So this knocks out the entire secretion system because it loses one critical component in that injection mechanism. So when it loses it, it's completely unable to infect the plants. The plants are healthy. Photos don't really show it here, but they're largely healthy. And they're unable to colonize the plant. They sort of persist on the surfaces and on the inside, but they really aren't able to cause disease. So this was the very first step. This is all knowledge from other Pseudomonas syringae. We're just replicating it in the lab. So the first sort of thing we tried was knocking out that effector that's involved in sucking out nutrients from the leaves. So we knocked out AVR-E1, and very nicely we saw a strong decrease. It's a 1,000-fold decrease or more in its ability to colonize the plant. And associated with that was a loss in its ability to cause disease as well. So unable to colonize, unable to cause disease. Then we took the library that we already had of all these effector knockouts, and we screened them on kiwifruit over a process of months. And we found 3 other effectors that are individually required quantitatively that contribute to colonization of PSA. So we found these 3. These, however, don't reduce the colonization enough to reduce symptomology. So you still get disease-like symptoms. So they have a quantitative contribution, but they can still cause a weaker form of disease. So obviously that's those 4 individually contributes. What about together? You know, effectors like working in groups, just like humans like working in groups, unfortunately. So we group them into what we call redundant effector groups, or REGs. So what we found is very nicely this large locus of 10 effectors on the bottom left there, those are the 10 effectors, that when you knock them all out together, you see a small reduction in virulence. When I say small, it's about 10-fold. So it is significant, but on the graph it looks small. So it's very important that we sort of understand collectively what contributes to immunity, into the causing the disease. So this reduction, again, doesn't cause enough of a reduction to remove symptomology, but there is a quantitative contribution. We lucked out because these were all sitting in what's known as the exchangeable effector locus, all 10 of them sitting right next to each other, making an easy target. We can just knock it all out all in one go, and so we understand it very nicely. However, a smaller subset, you can sort of see it labeled as the FEL, those 8 at the bottom. When you knock it out, it's still fully virulent. If you knock out the top 2, it's still fully virulent. You have to knock out all 10 or a subset of those 10, which we are starting to narrow down now, to get that small quantitative reduction in virulence. And then finally, another redundant effector group. There's a lot of work that went behind this. I'm not going to cover that because these are all in different locations throughout the PSA genome. So we had to go fishing using knowledge that we had from other pathogens and try and knock them out sequentially to figure out how are they contributing to virulence. And so we found another 5 vectors that work together as a group. So to summarize, 4 individually non-redundant effectors that contribute to virulence. One of them is a major contributor, AVRE in red, and 2 redundant effector groups, reg1 with 10 effectors and reg2 with 5 effectors. What does this all mean? It's just numbers and names at this point. So the key about this is that we are learning so many things through this process. One of the earliest things that we learned was that effectors have, so before this, there were a handful of other pathogens that had been characterized similarly in sort of a sporadic way. But this work, funding over 3 years, allowed us to sort of do it in a collected, really condensed, concentrated form on one pathogen. But interestingly, all the studies that have been done before were done on what are known as annual plants, so plants that have a lifetime that span usually a year, a season. And there are some studies on ones that are biennial, so over 2 seasons. But the problem with this is if you imagine a plant that has resistance genes, and we're talking about resistance here, where the plant's able to defend itself against a pathogen. For an annual plant, the resistance genes only need to get the plant over the line until it can reproduce, to flower, set seed, and sort of spread itself. So it has like a short window. It just needs to survive that gap. Perennial plants, kiwifruit, for example, the plants last for decades, 60, 70 years sometimes in orchards. So when this pathogen came into New Zealand and it destroyed the orchard, this was a huge tragic loss. These are growers that have plants in the orchards that they know. They're like family members, effectively. And so they go in and they see this tremendous loss to their orchards, plants that they've had for decades. It was a huge loss. But on the flip side of this, we know that there's kiwifruit out there that is resistant to PSA. PSA is from China, where there's a variety of kiwifruit species that are growing there, and there's a large variety of PSA3 growing there. And so some of these plants will have resistance genes that are durable, and we love that term, durable resistance, because it means that these are resistance genes that allow it to resist PSA and survive for decades. So that the lessons that we learn from this is essentially completely different. These are resistances that allow the plant to survive despite infection. So that's the first thing. It's that we understand the effectors have an additive role in virulence. This is something that we had hints of from other pathogens. We now have a model that we are developing further and further for a perennial plant host. And we have, so how do we then deploy it into resistance? So obviously these are virulence targets, effectors that target the plant and suppress its immunity. If we can identify how they're doing these things, we can try breeding out those susceptibility targets, because these plant proteins are Achilles heels for the plant. We can effectively take them out. We may be able to take them out, and so the effectors are then useless to the pathogen. And so if they lose these effectors, they'll automatically become less virulent. So it's a really durable system of breeding resistance into plants that hasn't really been deployed very well until, hopefully until now. The second thing that we were able to do was then use that knockout library, and this is in collaboration with some master's project funding that we got from Zespri. And this is work that a current PhD student, Lauren Hamara, is doing. She did it for her master's research in our lab. She deployed the same library and found 4 effectors that are recognized in a resistant variety of kiwifruit. So this is Kiwiberry. You may have seen it in supermarkets, spanning, I think it's only 2 weeks that they're available in supermarkets. But it's a small berry-sized fruit. And the plants happen to be resistant, completely resistant to PSA. So unlike the previous graphs where we're looking at a high red bar and it dropping, what we're looking for in this case is a low red bar increasing because it's losing resistance. And so it's growing a little bit more in kiwifruit. So as a result of that earlier funding, we were able to deploy this and find really interesting targets that we can directly feed into breeding programs to introduce these resistance genes into new cultivars. And the second project that we were able to do was looking at the LPS. So the LPS in orange on the image on the left there is a extracellular glycopolysaccharide that's produced by PSA and a large number of gram-negative bacteria. We identified a locus that determines the shape of the LPS and its sensitivity to these toxins, taylorsins. They're like phage-derived molecules. And so as a result, we were able to publish a paper that outlines strategies to maybe use taylorsins as a treatment in orchards for management, because currently it's just managed with copper treatments, but you can use taylorsins, as well as some future phage work. So it allowed us to establish two collaborations, one with University of Auckland, another with University of Otago, that are ongoing to this day. So to sort of summarize, that should say bioprotection out there, oh, sorry. But BA partnerships fundings means new science. So this is, we characterize a novel model pathogen and plant system, resistance breeding for new cultivars in the future, novel on-orchard control treatments, as well as the ongoing collaborations that we have now as a result of some funding that we got for the project very early in understanding PSA. Nami.